Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. I'm Yvonne Stapp for Science for the Public, and this is our program, Public Science. Today we welcome Dr. Alonso Ricardo. Dr. Ricardo is from Cali, Colombia, and he took his PhD at the University of Florida. There, his mentor was the distinguished biochemist Stephen Benner, one of the leaders of the development of artificial genetics. Dr. Ricardo is a research associate at the Szostak Lab at Harvard University, where he works closely with the Nobel Prize winner, Dr. Jack Szostak. Dr. Ricardo's pioneering research on self-replicating chemical systems addresses several fundamental questions. In this presentation, Dr. Ricardo explains how life emerged from inanimate matter and how RNA contributed to the formation of the early cell. He also explains the implications of the synthetic cell, the work in which he plays a very significant role. Dr. Ricardo's publications are very technical. However, he's written a very accessible and excellent article on the topic of this presentation in the September 2009 issue of Scientific American. We highly recommend that article, Ricardo and Szostak, September 2009. And you'll find the link on our website where you can click on resources and then life origins to see the title. And now it's my very great pleasure to welcome Dr. Alonso Ricardo.
Hi, today I'm going to tell you a little about what we know, the origins of life basically, and this problem on trying to understand how life appeared on our planet. As Yvonne introduced me, I'm Alonso Ricardo, and in the Szostak Laboratory at Harvard University. Today we're basically going to start with a little overview of the research that I'm doing. And so the lecture that I'm going to give today basically covers several topics. In the first part of the lecture, we're going to talk a little about what is life, and how can we define life in order to be able to understand it. In the second part, I will tell you a little about life's building blocks, their origin, where did they came from, the structures which are a little important in order to understand how also they interact with the rest of the matter. The third part, I will tell you a little about information transfer and catalysis in biological systems and why is this important. Last, we will discuss a little paradox, which is basically of chicken and eggs, and it's this problem that nucleic acids like RNA seem to be a little handicapped to perform some functions that are more suited for protein molecules, which are a big larger molecules, but at the same time proteins cannot exist without in modern cells without the participation of RNA. And last, I will tell you basically some of the more recent advances towards the construction of something that is synthetic life. It's not properly the life as we know it, but it's something more synthetic, something that we're doing in the lab. So the first part is basically what life is, and this is a hard question. This is something that is hard to define, and there are philosophers and people all over the place trying to really define what life is. But we don't want to get into the philosophical discussion on what life is. We need something practical to work with, and so what we're basically trying to do in our work is adopt what is called a operational definition, which is basically the definition that NASA has provided when they designed their missions to go to look for life elsewhere. And it's basically that life is a self-sustained chemical system capable of undergoing Darwinian evolution. This definition, because it's a definition created to things that not necessarily apply to terrestrial life therefore applies to things that are non-terrestrial, so also applies to non-terrestrial life, and in that sense is very global, and it contains a lot of information in that little sentence. For example, it says that life is a self-sustained chemical system. This implies that this chemical system, it needs to have a certain level of organization and certain properties that allow it to have that self-sustaining. And also it brings this little word, which is Darwinian evolution, which it carries a lot of luggage with itself, because basically it implies that this thing that we call life needs to have the ability of reproduce, and not just reproduce, but also have some little imperfections during the reproduction process, which will allow life itself to evolve, because these little imperfections will probably create some advantage to the living system over others that don't have that mutation. So that's the definition we're going to start with, and I just wanted to show you that modern life is very complex, and as we can see, it's obviously the product of several billion years of evolution. So here I had two pictures, one of a unicellular organism, which already looks pretty complex to me. If you look at it, it has a little membrane, it has some organelles inside, and it has a set of acquired properties that allows it to move, for example, in this case we're displaying this movement that you see in here. To other things that are present in multicellular organisms like us, which are cells that are a little more complex. And in here you have a white blood cell, for example, which is in charge of destroying entities that are strangers to the body. But we're not going to talk about the modern life as we know it today. We're a little more concerned about the origins of this life. So when did life start? So we don't really have an exact number to where the life started, but we can basically come up with a good estimate based on the fossil record. Of course, 3.5 billion years ago, we're not going to have any bones to find and characterize. In fact, there were no bones at that point because we were talking about organisms that were very simple. So the best information that we have is these structures called stromatolites, which some of them have been dated to be as old as 3.5 billion years. And what these stromatolites are basically the residues left by microorganisms that were living back then. And basically these microorganisms will leave sediments that are minerals, and they will make little biofilms on the surface of rocks where they were living. And basically you will see these formations in which you can clearly see several levels of deposits and layers of different structures that are basically minerals that over time had formed these characteristic structures. So there is some stromatolites out there, modern stromatolites, and they look like this, like the ones in this figure. The other evidence that we have are what is called these microfossils, which are basically little structures, as you can see in this figure, which are found in very old rocks. And basically people has been able to, by doing different studies with carbon isotopes, to show that these perhaps were actually originated by organic life. And basically these shapes correspond to things that look like this, which are some cyanobacteria that we had in modern life. So we can say fairly that life is old, and is probably in between 3.5 billion years ago is when we start having a record of life. All right, but what was the air like back then? Before this 3.5 billion years ago, when we had the formation of this forms of life. And I want to first acknowledge Janet Isawa, who is probably the person who created most of the animations which are going to be presented today, and some of the pictures. So the air back then, we're assuming that it had liquid water and that it had an atmosphere. And basically this is a nice picture that basically shows you all the possibilities that could have happened there. And we see an ocean, and we see a volcano with its emanating gases, which basically are gases that, because of the nature in which they are formed, will allow certain chemical reactions to happen spontaneously. Also, you see a meteor falling from the sky in this picture, and this correlates with what astronomers tell us had happened previously in our planet. So how complex was life when it started? So we, the people doing this research, and in a few minutes, hopefully you, the people trying to learn about these things, will assume that and will think that it was not. Basically, that life was a little more simpler. In order to basically understand how life works, how these modern systems behave, basically we need to try to see what life is made of and how these components interact with each other. So what we're going to try to see is basically what are the chemical building blocks of life, which if you want to draw a computer to an analogy to a computer, you will say hardware, and what are the informational molecules, which will be in this case the software. So with that, let's start with then the basic building blocks of life. So modern life basically is made of three components, a little more than three, but basically three are the main characteristic components, which are basically DNA, RNA, and protein. I say three because DNA and RNA are sort of relatives of each other. They just differ in a very subtle way in structure, but they are what we call nucleic acids. They perform different functions, and we're going to go over that later. So and the proteins, and then we had this whole family of compounds which are called lipids, and these lipids are the components of membranes. And in here you see a picture of a vesicle and a micelle. The vesicle has a content inside. The micelle doesn't have any content. The DNA and RNA basically are these two molecules which they can do several things. It's mostly related to the storage of genetic information. In the case of DNA, in RNA, it plays both roles. It can do the same functions that some proteins do, but also can take some of that information that is stored in DNA and move it around. The proteins are made of building blocks which are amino acids. We'll show you the structure later. And the DNA and RNA are made of basically two components, which are nucleotides and a molecule of orthophosphate or inorganic phosphate. Lipids, on the other hand, they come in two flavors. They are phospholipids, in which basically fatty acid molecule, which is shown in here, which is basically a long carbon chain with a little polar head, which is a carboxylic acid in the chemical jargon, is modified with a phosphate and a glycerol molecule to make phospholipids. So basically, fatty acids are components of phospholipids. So where did these molecules came from? So when we think about it, we think there is two possible ways that these molecules could have a rise to a planet, and one of them is exogenous sources, meaning of extraterrestrial origin, and the other one is endogenous sources, which means that they will made here on the Earth. So how do we scientists look for things that are of exogenous sources? So let's say you really want to find out if something came out of space and what is it, and how can I learn what is in this rock. So what we know is that there is certain meteors which are characterized as Carbonaceouschondrites, which basically just means that these meteors are rich in carbon molecules. And there is a few then who several people have been able to collect in conditions in which the contamination of terrestrial organic molecules is minimal. So some of these meteors had fallen, and people had been able to collect them, within a few hours of their stance here on the planet. And so when you get one of these pristine rocks, what you want to do is basically analyze the contents of this rock, the organic contents, and see what is inside. So what you do generally is that you take the rock and you break it apart, and you take the interior of the rock, which is the part that has been exposed the minimum amount of time to terrestrial conditions, weathering and water and microorganisms and all sorts of things. And basically do a chemical process in which you extract some of the components that are inside of that meteor, and then you put this mixture of components into a machine that is able to separating. This is the sort of machine that we use that is represented here, that is able to separate those components and then give you some kind of information, such as, for example, the molecular weight of one specific compound or many other properties of one specific compound. And then what you do is that you compare those results with standards of certain compounds that we know and that we can make here in the planet. So when you analyze these rocks, then basically you can get the information from the meteor and then say, look, we found this compound because it has the same characteristics as this other compound that we found in the planet. And then you can basically analyze this data and you can, interpret and say, what were the components that were present in that meteor. So what we have learned, how we learned so far from the analysis of these meteors. Well, the one thing is that these kinds of meteors are rich in the same kind of organic compounds that are found on living systems, basically, for example, amino acids. So amino acids are the components of proteins, and a large number of amino acids have been found on these meteors. Also, we have found components of nucleic acids such as DNA and RNA, and this is a molecule here depicted. This is adenine, which is one of the components. Is the A on your RNA. Your DNA basically has A, G, C, T. Those are the 4 bases, and adenine is one part of the A component of your DNA. So you will say, all right, they have found these compounds. What else have they found? Well, we have found sugars. Some other people have found fatty acids, which I told you can make these vesicles and can make these micelles. And some small amounts of phospholipids have also been found on these meteors. So the question that comes to mind is, all right, so how do we know that these meteors, these molecules you are finding, are not the product of just contamination, that perhaps, you didn't do a good job opening up the meteor and just getting the sample inside? And the one reason why we know these are not contamination is that people have found compounds that are not indigenous to our planet, basically compounds that are not that abundant in the planet Earth, and they're very abundant in these meteors. One particular case which is really interesting is these molecules here, which are called alpha-methyl amino acids. So the regular amino acids that you see here, they have a little hydrogen atom in this position, as you can see in here. This little white ball represents a hydrogen atom. What in here, in this other set of amino acids found in other type of meteors, you will find that hydrogen is now replaced with a carbon, which is attached to 3 other carbons. So these type of amino acids are very rare in our planet, and since they are so rich in these meteors, and we found them too, so we conclude that the ones that don't have the methyl and the ones that had the methyl were both present in the meteor sample. So it's a good way to basically, do a self check to see how good you're doing keeping your sample clean of contamination. So we know so far that there are components of proteins and of nucleic acids and also fatty acids that come from these meteors. Another question is how are these compounds formed in these meteors, and what type of chemical processes are required for these meteors to be formed? And so there's the second group of people which are basically studying something that is called astrochemistry, and it's basically chemistry that occurs in very strange conditions, which are conditions different from what we have in our planet, and they mostly occur in the gas phase. And they require things like ionizing radiation. These reactions can also occur in another planetary bodies such as Mars, the interstellar space, or in other bodies like comets, which are known to visit our planet quite frequently. So how do you know what is out there in space, for example? Let's say you're interested in seeing what is in a molecular cloud. How can you detect that? So people like Lucy Cirrus, which is a professor at Arizona, what they do is basically they go to a lab and they make a set of compounds and they simulate the conditions that you'll find in space they put these compounds under those conditions, and then they record a spectra, which is basically a sort of like a molecular picture of what the properties that reflects the properties of the molecule. So what they do is that they go to the lab, and they make the little gas phase simulation, and then they record this using these instruments. They record the thing that is the spectra. All right. So once they had now that spectra, and they know, for example, they take the molecule. They say they take glycolaldehyde, which is an important molecule for the origins of life. They go and they record this spectra in the lab, and then they go and use a radio telescope and go and basically look in the sky, and they focus in this molecular clouds, and then they scan these molecular clouds to see if they can find the signals that match the glycolaldehyde signals that were created in the lab. And so in that way they can basically, by doing this comparison, look to see if the same molecules that we know that are important for life are out there in space. And the answer is yes, and they have found all sorts of interesting molecules. In here I just have a few molecular pictures of some of them. But these molecules that they have found are in fact very important because they are building blocks for DNA, RNA, and proteins and lipids, and are things that if we take if we bring these molecules into a chemical reaction in water, we'll start getting things that look more and more like the molecules of life. So now let's talk a little about the endogenous sources. So basically, how are these compounds made also here on our planet? So this is what is a broader field, and this is what people call prebiotic chemistry. And so what are the conditions on the planet that are required for this chemistry to happen? And basically there is a few. So you require an atmosphere to prevent everything from escaping. The atmosphere could either be reducing, meaning that there is a high ratio of hydrogen in the organic molecules, basically like methane, like ammonia, which are molecules that contain an heteroatom, basically something that is no hydrogen and a lot of hydrogen attached to them. Or they can be oxidizing. Atmospheres can also be oxidizing, which is basically rich, for example, in something that has very little, basically no hydrogen, like carbon dioxide. In chemistry, you generally require, for interesting things to happen, you require an energy source, which in our planet it comes from basically having in the vicinity a star, which is the sun, and also from the volcanic energy that is inside of the core of our planet, and which comes from the radioactive decay of heavy elements that basically generate at the end of the day volcanic activity. So this is actually a picture here, what you see, which is quite amazing, that shows all the wonderful things that are happening during a volcanic explosion. And what you see in here is basically this is the volcano, and you see this cloud of gases that are coming out of the volcano, which are generally gas that are reduced, basically contain a high ratio of hydrogen to other atoms, and that allows that chemistry starts to happen, because now the hydrogen can get substituted by other things in the presence of, for example, molecular oxygen. And when we had this kind of like clouds, it's interesting that in volcanoes you also see this. I don't know if you can see the lightning in here, which are basically just generated during the volcanic explosion, which is another source of energy for chemistry to happen. So this brings back the idea of this famous Stanley Miller prebiotic soup, which is basically the primary experiments on prebiotic chemistry. There's one thing that we cannot forget about it, and also minerals. And so we don't only have all these organic molecules playing around, there is also a strong contribution of all the minerals present in our planet that may have either modulated or facilitated the chemistry that happened in order for us to get life as we know it today. So I mentioned Stanley Miller, so here's a picture of Stanley Miller, and this is the type of experiment that he was doing in his laboratory. So what he will do is basically just put in this little balloon here a mixture of gases In his case, he was using gases that were reducing gases, as I told you before, so basically things that are very rich in hydrogen. And then he will create an electric discharge between these two electrodes that you see in here. He's here providing the energy to one of the electrodes. And what he will get will be as, the gases will react and they will fall back. Whatever molecules were being formed, they will basically, there was a flux of water, they will fall back in here and get dissolved in the water. And then he will treat this water with acid and he will analyze the contents. And what he found is that you could actually make some amino acids. And not only one amino acid, you make a large and diverse set of amino acids. And as we know, amino acids are the building blocks of proteins. Some people say, well, the experiments that Miller was doing with reducing atmosphere. So we know today that perhaps the atmosphere back then was not so reducing, that things were a little more oxidized. But other people had tried similar experiments, and at the end of the day, you can always get some amino acids with this type of conditions, even if the atmosphere is not so reducing. So what else have we learned after all this wonderful work by Miller? There has been a lot of work by many people, and one of the most recent work was done by John Sutherland and Matt Pauner and others at the University of Manchester in England. And what they found, it was basically this concept that generally people try to separate the chemistry of different things. So for example, people will try to put the chemistry of the nitrogen in one side and the chemistry of the oxygen on the other side, because when you just mix things, the random things start to happen and you get something that resembles more tar than anything that resembles life. But what they found is that under certain conditions and in the presence of phosphate, you could actually mix these two chemistries, the chemistry of nitrogen-rich compounds with the chemistry of oxygen-rich compounds, to make something that then will react later to make a nucleoside, which is one of the building blocks of RNA. So this was a great discovery, and I think it now has provided a lot more clues about how all this chemistry came together. And for the first time, it shows us that we cannot really just exclude things, but actually makes more sense to put everything together and try to see what something like phosphate, for example, which is an inorganic compound, could do to these molecules to allow them to react in a way which actually leads to the formation of something that resembles life. So this nucleotide that is represented here, that was created by in the Sutherland group, has an interesting thing, and it has this cyclic phosphate. And in chemistry, when you had things that are cyclic, you can store energy in them, because basically the molecule is constrained, and that energy allow us to call this compound to be activated in a sense. So basically meaning that this component will thermodynamically undergo a reaction under the right set of conditions. So to show you a little of what these activated nucleotides can do, I will show you an animation in which will show what nucleotides do when they're put in a context that allows them to react, for example, in a clay mineral. And so some people had actually taken activated nucleotides and put them on clay, and what they had seen is that these nucleotides polymerize. Basically, they start joining together. So here in the animation, what you will see, this here represents basically a clay mineral surface, and what you see here is the little nucleotides that are floating around, and they start to get attracted by the metal on the clay, and they interact with the phosphate on the nucleotide, and they start aligning, next to the clay. And this is just a model of what we think is happening. And then when they're aligned, because they're so proximal to each other, they start to react. And what you see here are little, what are called leaving groups, which are basically allowing the bond formation process to happen. So what we think is that, you can get these minerals which are abundant, and if you had the right set of activated nucleotides, they could actually interact with this mineral and start making chemical bonds to make structures that look like DNA and like RNA, for example. Now let's move away a little from the nucleic acids and let's talk about phospholipids and fatty acids. So as I told you before, phospholipids are made of glycerol, fatty acids, and orthophosphate. Basically, they contain a hydrophilic head, basically something that likes water, and a hydrophobic tail. The fatty acids are a component of the phospholipids, and we know that fatty acids are known to be synthesized in hydrothermal vents under conditions of high pressure and high temperature. But the one interesting thing that is also present is that fatty acids can form vesicles. So this is important for several reasons. First, the fact that you can get fatty acids directly from a thermal process, like in a hydrothermal vent, tells you that these materials were readily available, given if the carbon source was present. And we know that was the case. So fatty acids are definitely one of the things that we know that could be formed in the early air. And now, since fatty acids had this property, they can make little vesicles, then we could start thinking about fatty acids as being a primordial component of basically a cell in which phospholipids were not present, but fatty acids serve as the compartment for this cell. So how are these fatty acids formed? So in a hydrothermal vent, if you think about it, we're going to just take a snapshot inside of the hydrothermal vent, and we're going to have conditions of high pressure and high temperature. And this process is known as a Fischer-Tropsch type process, in which what you see in here is the formation of a hydrocarbon chain. So basically carbon gets reduced with hydrogen, and it starts making these little structures that you see in here that are elongating, and they just keep growing. Sometimes water gets into it, and they can also get oxidized, and then you will get your little fatty acid particle. Then they basically become detached from the surface in which they are being formed, become suspended in water, and then when the hydrothermal vent expands of a geyser, basically just spill them out, you will have droplets of water that contain the fatty acids attached to them. The polar head, basically the carboxylic portion of the fatty acid, will be interacting with the water, and the other one will be trying to repel the water. These water droplets could fly away during this evaporation, and then you could also have some little micelles being formed in the process, as is shown here. So this could land in a different location, and water slowly evaporates, and you will enrich this fatty acid concentration. Now, what happens when you basically enrich the concentration? Well, when you put a lot of fatty acids together, they start making initially micelles, but then when these micelles start interacting, here's a little video that I show you. When these micelles start interacting with each other, if the concentration is high enough, they start making initially something that looks like a monolayer, basically of fatty acids, in which the tails of the fatty acids that are hydrophobic are trying to get together, and the polar heads, which like water, are outside. And basically, if the concentration is high enough, these fatty acids will start moving until they make these little structures, which now can actually have something inside of them, and they're called vesicles. So that's how they could potentially be organized, and how they could have formed a primordial membrane, basically, if you want to call it that. But what is so important to have a membrane? Why do we need a membrane? Why do we care so much about lipids? And the reason is that they provide something called compartmentalization, and which is very important. And in this animation, I will show you why. So imagine that you have a molecule, basically an RNA molecule, that has learned a trick. And this trick that the molecule has learned is that it has learned to make copies of itself. It became a little Xerox machine in which can make copies of itself, but can also make copies of other things. So in the animation, we'll see a little more clearly here. So this little box here represents the RNA molecule, or what we're going to call a replicase. So when this replicase finds another molecule of itself, it will make a copy. So now you had several copies of the replicase being made. Now if this replicase finds other thing that doesn't quite look like it, it will also make a copy of it. And now you will have, things that are not that useful because those little balls were not replicates. Now imagine there is a pool of molecules that don't have replicase properties. So the replicase will start making copies of everything, every single one of these things. And at the end you will end up with a bunch of useless molecules, and nothing much is going to happen after that. But if you put a compartment, in this case we put a little vesicle in here, the replicase will start only making copies of itself and will quickly fill, and you will have multiple copies of the same molecule. So that's important because that's what provides the driving force for in some cases evolution, that you have this compartmentalization and an advantage. In this case, the ability to replicate will be kept isolated, and then the molecule can evolve further functions. All right. So that's about, we talked about already about the little nucleic acids, what are they important. Now let's talk about proteins. So modern life contains proteins, which are the moderators and accelerators of cellular processes. That highly specialized molecules. They perform very specific functions, and they, as you can see in these pictures, they're large ordered structures. Some of them, for example, this structure here, they tend to basically insert themselves into membranes, and they allow the passage of certain things. Phospholipid membranes are very tight, and not really anything can go in and out, except things that go in and out via the participation of protein molecules. That's actually I forgot to mention, that's one of the advantages of the fatty acid vesicles compared to phospholipids, and is that fatty acid vesicles in fact allow things to go in and out because they are not as rigid as the phospholipid vesicles, which had been an advantage in a prebiotic world. All right, so going back to the proteins, the proteins are are very interesting. They're very large. They contain hundreds of amino acid residues, and they fall into these very nice and three-dimensional shapes. Proteins help to replicate nucleic acids, but nucleic acids contain the information required to make the protein. So proteins haven't learned the trick yet of being able to make copies of themselves de novo, basically just take amino acids and start making a copy of itself. So that's bring us again to the nucleic acid. So nucleic acid seem to have all these interesting properties. And if you think about a nucleic acids, you can simply think in the way they interact, in the way they transfer their information, as if they were a Lego set. So in here we had the two most dominant pairs of nucleic acids in life, which are guanosine, cytidine, adenosine, and thymidine. The way they're drawn here is what we call the Watson and Crick base pairing sets, in which guanosine base pairs to G and adenosine base pairs to thymidine. If you look closely, these base pairs are basically what we call hydrogen bonds, in which a highly electronegative atom is basically donating the electrons to a hydrogen, which is attached to another highly electronegative atom. So basically the hydrogen his electrons are being taken away, and he's trying to reach for any electrons he can find, and basically these nucleotides have this ability of interacting by donating the electron density to the hydrogen that is depleted of it. So the analogy with the Lego comes from the way that they can interact. For example, guanosine and cytidine make 3 base pairs, and when you see here, these represent these protrusions here represent the hydrogens, and this will represent the electron donors. So in guanosine and in cytidine, you have basically 3 pairs of possible interactions. In adenosine and thymidine, you had these 2 sets, which is what we see. Adenosine doesn't have a hydrogen here to share with this other set of electrons in thymidine or in uridine in the case of RNA. But what we know is that if we put it if we create synthetically something here that can match in here, they will actually do it. They will match, and so this is a very simple way of looking at the design of these nucleic acids when you want to make modifications. So nucleic acids are, as I said before, are the molecules responsible for the storage of information required for cell function and replication. And the information of the nucleic acids is encoded in the linear sequence of a nucleic acid polymer. So basically the components, the way that the nucleic acid has organized A, G, C, and T, or A, G, C, and U in the case of RNA, is what dictates what information is inside the molecule, what event in the cell is going to happen. As I said before, there is a very tiny difference, which is a very tiny chemical difference, but structurally has a major consequence, and is that RNA has this hydroxyl group in here, and DNA has only a hydrogen group in here. This is the sugar portion of the nucleic acid, and what it does is that it changes a little the geometry of the molecule by just having that substitution, and also it changes the stability, because this oxygen here can participate in chemical reactions that cleave RNA by itself, while in DNA this hydrogen cannot participate in those reactions. So for that reason, DNA is a little more stable in water towards hydrolysis than RNA. So if you're an evolving organism, it will be an advantage for you to have DNA as to store your genetic information, because basically you can store it for a longer period of time. With that in mind, but you will get the picture that, yeah, nucleic acids are these just molecules that just sit around and store information and move it from one place to the other, but in fact that's not the case, and that's what is so interesting about them. And RNA, for example, we know now that it's able to form complex three-dimensional structures, and what you see here are basically pictures of RNA, and this is the ribosome basically. And what you see is that the majority of the ribosome is basically made of RNA. So in fact, RNA can fold. If you remove the proteins, what you get is this. RNA can fold just like a protein, and in fact, it performs catalytic functions just like proteins do. So in fact, the ribosome active site, the place where new proteins are made, the key residues that make the peptide bonds that make the proteins are all made of RNA. So RNA is a the ribosome is basically what we call a ribozyme, or a catalytic piece of RNA that is able to achieve chemical reactions. So we know that RNA can act like a protein. So now this is interesting, because if RNA can act like a protein and also can act as a nucleic acid to store the information, so perhaps we didn't need, when life was just starting, we didn't need proteins at all, and basically we just needed a molecule that was able to do the two things. So RNA can store information just as DNA, can perform catalysis just as protein. RNA can also perform complex molecular recognition. I told you that these complex forms of proteins allow them to basically go and find a specific molecule and bind tightly to it, and RNA can do the same thing. The other thing that RNA can do is then can mutate, and under the correct selective pressure can also evolve. So the question is, could life have started with an RNA molecule that was able to catalyze its own replication? Which it will look somehow like this. So imagine now that you're in the primitive world and you had this RNA molecule that folds, and now it's able to find another RNA molecule and start making a copy of that RNA molecule. If this extended molecule has the same sequence as this one that just flew out, if you hit this new duplex, it will eventually denature. Basically, the hydrogen bonds start coming apart, and the two molecules come apart as the heat increases. And now, if these molecules fall into the correct conformation, you will have another replicase to undergo again another round of amplification. So That's the sort of thing that we imagine that could have happened in the early life, in the early years. So how do we know that this is in fact how it happened? So the way to do it and the way that we approach this problem is try to make it in a simple way. So the way we're trying to do it is basically trying to construct what we call a protocell, which is basically a minimalistic version of a cell that has the components that we think are important to be considered a cell. Basically, what we think it must have is basically a membrane compartment, which is going to be made of fatty acids in this case, because are the molecules that we think they were more likely available back then. And it will have inside a nucleic acid component, which will be able to feed from molecules that come from outside and perform catalytic functions inside, such as replication, that will allow and slowly evolve and gain properties that will allow this cell, to gain new abilities and then start evolving furthermore. But the question is where to start. Where do you start when you want to make something like this, like a protocell, something that mimics the primitive cell? So we know that lipids, basically fatty acids, we don't really have to do much with them because we know that they self-assemble. They will spontaneously self-assemble into vesicles. So if you put these fatty acids in water, they will start making these sort of structures that you see in here. But then what we need, the second thing that we need once we have this is basically a replicase. And when I mean a replicase, we mean a nucleic acid, hopefully something that looks like RNA molecule that can use molecules or food sources, let's call it food sources, that are coming from outside, and use that to make copies of itself or to perform different kinds of functions. So there's two ways of doing this, and the first way is the one that the Szostak Lab approached in early 90s, was basically try to evolve in a test tube an RNA molecule that will have this activity. So what they did basically was that they created a large library of RNA molecules, which was made of billions of components. And then they basically applied some sort of selective or evolutionary pressure to get only those molecules that will display a desired property. In this case, those molecules that were able to take nucleotides and incorporate them into a growing molecule. They will put little tags in these nucleotides to make them distinguishable from itself, and then they will collect those molecules that display that activity and repeat the process. So basically they were doing what we call an in vitro selection evolution experiment. What they found is that it works. So they got something that was able to take nucleotides, take another molecule of RNA, and start making a copy of that molecule of RNA. So the problem was that what they found was a polymerase, was basically an RNA molecule that is able to take pieces and put them in front of the other one. But unfortunately, it was not able to finish these copies to make a copy of itself. It just made little stretches of nucleotides. The one problem that it had is that it was slow. It had very little rates of catalysis, and so therefore it will be prone to hydrolysis by water, because, as I say, RNA is not too stable in water over long periods of time in the presence of metals. However, this is by no means saying you're not going to be able to find it. The thing is that this was done in the periods of weeks, and the large sequence space that is present in molecular space is too large to be explored in the settings of a laboratory, for example. So especially the people doing this are grad students, so you really need to move on and you need to find what works for you. And in this case, the large sequence space needed to be explored for this, for finding something like a replicase. It's just too large and we don't have the technical ability to do it now. So is there any other way to obtain a self-replicating nucleic acid molecule? Well, then it becomes the alternative. So now instead of having an RNA molecule that folds and makes a copy of itself, what if you could take nucleotides and modify them, basically the building blocks of the nucleic acid, modify them in such a way that they could organize themselves and then react to make copies of other things. So this is what I show here in this little animation. And basically in this animation what you see is a piece of nucleic acid in which now the nucleotides were designing nucleotides that are able to find the complement nucleic acid and align themselves on it and then react. The interesting thing about this is how we put it into the animation, and the animation was actually done before the experiment, but this was the conceptualization of what we wanted to happen. So basically what we do is we go to the lab and we try to use all the knowledge that we have, basically all this knowledge that G only pairs with C and that A only pairs with T, and we try to fine tune those nucleotides in order to achieve that base pairing, and then that base pairing will drive the chemical reaction, the next chemical reaction to extend the polymer chain. So this is the same sort of representation of what we need to do, and basically what I'm showing here is this is the template, what we call the template strand. It's basically a piece of RNA which we want to make a copy of, this one in here. And this is what we call a primer, which is just a little piece of nucleic acid that is complementary to the template. And then these are the monomers that we add. So what we think is going to happen is that these little monomers are going to align. So G is going to align in front of C. This other G is getting close to this other C. And once they start aligning, they start reacting because the chemical groups at this end will spontaneously react. And for example, in here, this is the first step. You see this U is attacking this cytidine here, and now this bond is formed. And now this C will attack this G here, and this bond will also be formed. And this becomes sort of like a chain reaction. And at the end of the day, you'll have your full copy by just adding monomers. So you don't need a replicase molecule to be able to make a copy of another molecule. And so how do we look at this thing in the lab? So what we do is that we take this little molecule here, the primer, and we put a label on it. In this case, we put a fluorescent label, and then we do something that is called an electrophoresis experiment, in which we basically separate these molecules based on their size. So the primer, which is short, is represented here by this dark spot. This is just the primer by itself, and as the primer grows, we add then the nucleotides, and as the primer grows, you start seeing the appearance of other bands here. And these little darker bands, what it means is that the primer is getting extended. So basically it's getting a little longer and longer over time. What you see in the experiments that we're doing is that after 6 hours, we already had full copying of the template. And if we leave it for longer, just basically it finishes up everything that it was in between. So this was very exciting, and it was the first time that anyone can show that this is actually feasible, that you could get these mechanisms in which you can basically make a copy of a nucleic acid molecule just based on the chemical energy of the nucleotides and stop where you're supposed to stop, which in this case was when the template was completed. The second step was basically try to see if this will work in a vesicle. So the experiment that is shown in here, this other part of the gel to the right here, is basically what we do in there is that we take the primer and the template and we encapsulate it inside a vesicle. Now this primer and template inside the vesicle, we add the nucleotides outside of the vesicle. So these nucleotides had to go inside of the vesicle and then react, basically find the primer and the template and then react to finish up. And as we can see, it takes a little longer. It's not as fast as before. So at 6 hours, we don't have full completion. But after 24 hours, now we see that these monomers are able to basically get in the water, cross through the little vesicle, and inside undergo this chain reaction to basically make a copy of the polymer, this is sort of like a proof of concept of what we want to do with a protocell. It's not a real protocell yet because we haven't achieved self-replication yet. Basically, we have not demonstrated that we can basically just mix these things and they will start making copies of each other right away. But we're working in the process, and so far it's looking very good. There is a lot of synthetic chemistry that it needs to be done in this process. These molecules are very different chemically from the natural nucleosides, and are obviously not natural. They're unnatural nucleosides, so we had to spend a lot of time making them, and that has taken a little bit of time for us. So now the question is, okay, so once you have a nucleic acid system that is able to, undergo this chemical replication, and it relies on external sources of food, in this case activated nucleotides, will they be able to, as the copies of the nucleic acid inside of the vesicle start being made, and the division you need to at some point to divide, you need to have daughters. In this case, the protocell needs to grow and divide, and each part of the protocell will divide with a little portion of the nucleic acid. And now, because these nucleic acids are in different protocells, they don't need to behave the same way, so they may mutate, and these nucleic acids in this protocell may have different properties than these ones over time. So it's important to have a mechanism in which you can basically divide the protocell. What people had done in the past was basically take vesicles and extrude them through very tiny little pores. So basically, if you have a large vesicle and you pass it through a little pore, what's going to happen is that this vesicle, in order to pass through that pore, is going to rupture. So you'll make little vesicles by doing that process. But so a grad student in the Szostak Lab, whose name is Tinshu, this very interesting experiment, and what he did is that he discovered this rare mechanism. It's actually not rare, it's quite common, but it was unknown of. And what he noticed was that when he took a vesicle and he added little micelles, as you see in here, the vesicle will grow this little tail, and then the vesicle will start growing into these elongated shapes, as you see in here. And then just by agitation or just by blowing some air through it, this thing will divide into little components. And then he will repeat the cycle, and then basically you could couple the division of these vesicles with the addition of micelles. So this is very interesting because this is something that you can actually imagine happening in a prebiotic planet, when you had your vesicle that contained a genetic material, and this vesicle then more fatty acids components started arriving to this place. This vesicle starts growing with the genetic material inside, then elongates and then divides with the material. So, but how did actually Tim look at this experiment? So what he did, and I have he gave me a movie to show this process, and what you see in here is the little initial vesicle, which has a nucleic acid component that has been labeled with a fluorescent dye, and that's why you can see it. What you can see is that as he starts adding micelles, this vesicle now grows into this filamentous shape that you see in there. This is actually a real tiny, it's hard to see sometimes, but what you see is these elongated shapes in here. And now these elongated shapes, when he provides an external source of force like air or just basically movement, they will divide into daughter vesicles. So you see them here, he's moving the microscope focus, but now you start seeing that they started already breaking apart, and where there was only a single original vesicle, now you see a lot more of those. You see at least 3 or 4 or 5 daughter vesicles that came out from this one. The important thing, and don't forget about this, is that inside of this vesicle there was a nucleic acid, and that nucleic acid was labeled with a fluorescent molecule. The fact that we see that the daughter vesicles are also fluorescent means that the division of these vesicles is consistent with the nucleic acid being transferred to the daughter vesicles, which is what we want to happen. So this is very exciting, and now we're looking into coupling these two things, this observation with the idea of having a chemical replication system. So at the end, what we envision is something that looks like this, basically a molecule, in this case a protocell, which is a protocell which is basically making copies of itself inside with these nucleic acid components, and it's also growing outside by the presence of the fatty acids. And at some point, because everything is, the food is getting there to make more copies of itself, and the lipids are getting there to get elongated shapes, it will divide, and now you will have two daughter basically vesicles in which each of them will have the genetic component that is necessary for continuing the evolution. So the current status is, what do we have so far? And so far we had a nucleic acid system that is viable for the study of self-replication. We have a physical process for the growth and division of vesicles, and we had lots of understandings about the chemistry, and what you learn actually a lot doing this is the beauty of life, is the beauty of how these molecules interact and how fine-tuned after so many billions of years of evolution they have become. What do we need to learn is basically, if the nucleic acid system replicates, are the copies similar to the parent? Are we going to have enough fidelity in our nucleic acid system to resemble the parent? Basically, also, if the time scales for replication and vesicle division are similar, and we think that's the case, and basically all of the rest, which I think is probably 99.9% of the things, because we really need to learn a lot more about this process. So I would like to acknowledge the Science for the Public organization, and in particular Yvonne, for inviting me today to talk about my research. The Szostak Laboratory, which is a great place to do science, and Jack has a lot of interest for all these origins of life problems. Janet Iwasa, who provided all the animations that I showed today. Tin Shu, who provided the movie and the figure that represented the protocell division. And I thank you for watching this, and thank you very much.